Hello everyone, and welcome to today's live broadcast, The Escalating Challenges of Antimicrobial Resistance and the Efforts to Conquer These Threats, presented by Dr. Dr Tillotson, a consultant medical microbiologist. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking site and producer of educational events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located at the lower left of your presentation window and type your question into the box that appears on that screen. We'll answer as many questions as we can at the end of the presentation. Also, please notice that you will be viewing this presentation in a slide window. To enlarge that window, just click on the screen icon located at the lower right. If you have any trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left-hand corner and follow the process for obtaining your credits. Without further ado, I'd like to now welcome Dr. Dr Tillotson. I will now turn the presentation over to him. Welcome, sir.
Hello, I'm Dr Tillotson. I'd like to present my perspective on antibiotic resistance and the escalating challenges that we're seeing from a clinical perspective with regard to emerging resistance and how are we responding to these challenges. So what I'd like to do is set the scene, give you a little bit of background, some of the complexities that we're facing with regard to antibiotic resistance, and then run through some of the unusual approaches that are being adopted at the moment. Whether they'll be successful or not, let's wait and see. But I've chosen about 7 different projects that I think will give you a feel for the breadth of activities and, dare I say, ingenuity that's currently being employed to try to defeat the ever-evolving bacteria. So let's move along. I just want to declare my disclosures. I'm actually a paid consultant for the companies listed on the screen, and none of them have actually paid for my time to present to you today, nor have I received any other form of funding. For those of you who recognize him, this is Sir Alexander Fleming, who did his momentous discovery in 1928. It took about 14 years for penicillin to actually be manufactured, but nevertheless, this is Alexander Fleming, and since 1928 we have, as I put it here, managed to destroy pretty much this man's work. And I think we're all used to seeing these sorts of headlines, whether it's on printed media, TV, and so forth. When the drugs don't work, the rise of antibiotic resistance. This actually was a front cover from The Economist last year, but nevertheless, the rise of resistance has continued, and I think the story of the problems due to these multi-drug resistant pathogens gets worse almost by the day. And I think it's important to realize that the problem is getting worse, and we don't seem to be learning a great deal of how to manage it. And the unfortunate part is that really since the mid-1980s, we haven't actually discovered any new classes of antibiotic. So we've been working along the same old classes. By that I mean beta-lactams such as penicillin, aminoglycosides such as gentamicin. Those drugs have been around for 50 years. Erythromycin, another very commonly used drug as a macrolide, first came into use in about 1953. So we really, we saw the heyday in the 1950s and 60s, but since the mid-1980s we haven't seen any new distinct classes of antibiotic. And what I'm going to do through the presentation is actually describe to you some of the new potential classes that I think will help us see some bars to this histogram going forward into the 2020s. So the bacteria have taken advantage of our lack of discovery and they are, as you undoubtedly know, extremely adept at finding new ways of defeating our efforts to control them. So, in terms of talking about control, the World Health Organization earlier on in 2017 actually issued what they call the priority pathogens list, and they broke them into critical, high and medium sort of priorities, and I've only shown you the critical and high here because I think that's where the bulk of the effort is really being focused at the present. And in fact, I think it's fair to say that in fact those in the critical box, the Acinetobacter baumannii, which is carbapenem resistant, Pseudomonas aeruginosa, also carbapenem resistant, and then the Enterobacteriaceae, which is a broad tribe of bacteria, and it comprises of Escherichia coli, Klebsiella pneumoniae, Enterobacters, and a range of other species, genera and species. And they are carbapenem resistant, as well as resistant to some of the more potent beta-lactam antibiotics, known here as third generation cephalosporins. So those are the critical targets for our current research efforts. But in the yellow box, there are still some pathogens that have hit the headlines quite recently. And in fact, the bottom line of that particular box, Neisseria gonorrhoeae, was actually the focus of quite a lot of WHO, United Nations, and other learned groups, their focus, because actually I believe it was in Spain where a strain of Neisseria gonorrhoeae was actually resistant to every normally used antibiotic. So although it isn't life-threatening, this is a major public health risk, and gonorrhea is back in the headlines, which is unfortunate. So that's the priority list. You'll have seen similar sorts of lists a couple of years ago from the CDC. Broadly, they're very similar, just the priority, just the listing has changed. So that's the WHO priority list. This is an image taken from the CDC website where they're actually looking at how do we prevent antibiotic resistance. And I think the top three are sort of good medical practice. We want to prevent the spread of resistance therefore we do that through preventing infections. Lots of good ways of doing that, washing hands and so forth. Good ways of preventing infections. I think one of the most important things is actually knowing what's going on, where it's going on, and sometimes how it's going on. So that's tracking. And there are some quite sophisticated tracking systems now. The US, obviously, the CDC system. Europe, we have what's called ECDC, and there are other systems around the world. But one of the things that I think is perhaps missing, dare I say, at the moment, is the actual intercommunication of these systems. And because we can all travel globally so much more easily than we could 10 years ago, 20 years ago, I think it's important for us in the United States to actually understand what's going on in Southeast Asia, Central America, and so forth, because we can all hop on a plane and within a few hours we could have taken or acquired a multi-drug resistant pathogen. So I think the tracking on a global level is really important, and I think the WHO is focusing on how for us to communicate better with each other. The other component that's gaining a lot of support, and again goes back to good medical practice, is that of antibiotic stewardship or prescribing. Now, the essence of antibiotic stewardship is to provide the best clinical outcomes without doing harm to the patient, but equally avoiding resistance development. And that, I think, has been the challenge, is how do we avoid resistance development? But stewardship in the United States has taken a twist in the last 12 months, in that one of the government bodies has now mandated that every hospital in the country has to have a stewardship program. So we're going to be seeing 4,000 points of care, all not having the same stewardship program, but very, very similar. So at least there will be a coordination, so to speak. So those are the three things that I think are core activities. And the fourth one, developing new drugs and new diagnostics, is part of where I focus. Diagnostics is has been a sort of a holy grail. We've always wanted a test that you can apply at the point of care, whether it's in a primary care physician's office or whether it's at the bedside in the intensive care unit. We've always wanted to have a test that we can take a bodily fluid or sample, rub it on a card or do something with it, and within minutes know what the pathogen is and maybe what the resistance profile is. We're getting there. Is it ideal? No. Should we be there in 5 years? I hope so. There are lots of logistical questions and, dare I say, economic questions. So until we have better diagnostics, which will in turn drive better stewardship, we're clearly looking for new antibiotics, new drugs. And that's what I'm going to expand upon going forward. So where are we today? Well, the table that you can see in front of you is reasonably comprehensive. I'm not claiming that every single drug that is in development as an antibiotic is on there. No, it clearly isn't. The preclinical column, the second column in, is sparsely populated, but you will see in a few moments that in fact some of the drug classes don't apply anymore because we're looking at new ways of trying to outsmart the bacteria. But if you go across phase 1, phase 2, phase 3, you will see that certain classes have been quite a focus of attention. And you'll see here, the quinolones right the way across the middle. There's been a lot of effort into the fluoroquinolone research, such that in fact a fluoroquinolone was approved by the FDA only 8 weeks ago, dalafloxacin. And that was the first oral IV drug to be approved in the US for probably about 5 years. So, there is activity. Is it the sea change that we're looking for? Maybe, maybe not. But you will notice that in that fourth column, the phase 3/approved, you will see there are beta-lactam, beta-lactamase inhibitors. That's the meropenem, vaborbactam, which actually is currently under FDA review, and we should know within the next week, I guess, whether this has been approved. And then you've got other drugs, new aminoglycosides like plasmicin, new tetracyclines, and I'm sure all of you have had a tetracycline at some stage. Those drugs have been around for more than 50 years. These are new derivatives. These are new glycylcyclines, so it's the tetracycline core with some modifications. And these drugs have got much broader spectrum of activity, and obviously they are effective against tetracycline-resistant strains as well as other resistance as well. I mentioned the quinolones. I think the quinolones have been one of the most widely prescribed antibiotics for the last 20 years, and you can see that there is activity at all different phases through with that class. The macrolides, those focus on 50S ribosomal protein synthesis, and there's been focus here, 3 drugs, solithromycin, naphthromycin, and lefamulin. Apologies about that. Solithromycin was actually not approved by the agency at the tail end of last year. They want some more safety data, and the other 2 drugs are in phase 3 clinical development. The folate antagonist, icloprim, it's based upon trimethoprim. Trimethoprim was a compound that was used quite widely in the late 50s, early 60s, and very effective for urinary infections, but sadly E. coli and its brethren learnt to outsmart that one quite quickly. So icloprim is likely to be effective against those types of pathogens, as well as MRSA, methicillin-resistant Staph aureus. I put C. difficile here mainly because it was on the CDC's list of bad bugs. Doesn't quite make it in the top categories for the WHO, but C. diff is still a major public health threat, still the #1 cause of gastrointestinal hospital-acquired infections, about 500,000 cases a year in the US alone. So there's been quite a bit of activity in this field over the last few years, and with some new classes being studied here, both phase 1, 2 and 3. So with a bit of luck, we'll be able to find ways to manage C. diff. And then on the bottom line, you've got some completely new approaches that are in preclinical, which is a The spiro compound is what they call a potentiator. That's an interesting way of looking at how to improve antibiotic use. The hybrid, the TD compound, is, if I remember rightly, a glycopeptide and a cephalosporin. And then gepotocin, I always get my tongue tied with that, but gepotocin, a new compound from GSK, which works at similar sites to the quinolones but is different, and it'll be interesting to see how that compound progresses. So I've given you a couple of moment overview in terms of what's going on mainstream, and I think it's good now to think about what are the other compounds and agents that are around. Not surprisingly, the bacteria have figured out multiple ways of avoiding antibiotic efforts, and I'm not going to go into this in depth, but some of the things that I've just mentioned, like cell wall synthesis, nucleic acid or DNA gyrase activities, folates, which is trimethoprim, these are things that we all know and understand. But I think some of the things that I'm going to speak about refer to different parts of protein synthesis and different parts of cell membrane ruptures. So there are ways in which these resistance mechanisms are going to be tweaked and challenged by some of our new activities. I'd like to focus a little bit on the cell wall synthesis approach to bacterial resistance, mainly because penicillin was the first antibiotic, and in fact drugs like ampicillin and amoxicillin are still the biggest, it's the biggest usage of antibiotics in many parts of the world. Ampicillin is still the #1 in India and many parts of the world. So just to give you a feel for how do the bacteria circumvent beta-lactams, and you'll have heard of multi-drug resistant E. coli and so forth. Well, multi-drug resistance usually means that the bacteria possess multiple ways of overcoming beta-lactams, aminoglycosides, tetracyclines. And you can see all of the major classes actually listed in the little boxes around here. But let's focus on the beta-lactams for a moment. There are three main ways, and this is not exclusive, and this is not totally comprehensive, but the three main ways in which bacteria evade our best efforts. One is enzymatic destruction or deactivation of a drug, and beta-lactams is a good example, and aminoglycosides are another example whereby the bacteria are able to add on chemical moieties that just deactivate the gentamicin, for example. Then you've got cell wall access. As you all well know, the bacteria needs nutrients to go in and out and various other chemicals. Well, the porins are the doorways. That's what allows these chemicals to enter to the cell. The bacteria are able to alter the size and shape or the number of those porins, those molecular doors, for want of a better word. And I think that is a very efficient way for bacteria to restrict the entry of different chemicals. And then the third one here is what's called efflux. And bacteria, like most living things, produce bio waste, shall we say, and they have to pump out its housekeeping. They need to export various waste materials, metabolites that they don't need, that may eventually be toxic. So they have this efflux mechanism, and I'll talk about the efflux towards the end of my presentation. But I think efflux is one whereby that process can actually take out multiple types of antibiotics with the same mechanism. So this is where multi-drug resistance becomes a real issue with one relatively simple protein change. So what are we doing on the beta-lactam front? Because it's the predominantly used antimicrobial class. What are the efforts that are going on at the moment? Well, if bacteria produce beta-lactamases, which is how they deactivate or destroy the beta-lactam antibiotics, beta-lactamases have been around probably for the best part of 30 years. The first one of which was clavulanic acid. It was combined with amoxicillin to make a drug that I think everybody may have heard of called Augmentin. So amoxicillin clavulanic acid. Bacteria have found ways of overcoming clavulanic acid, and in fact we were aware of that sometime 10-15 years after Augmentin, and piperacillin tazobactam was developed. And that was very effective against many of the multi-drug resistant hospital beta-lactamase producing gram-negatives such as E. coli. But again, the bacteria work out ways of overcoming what the BLIs, the beta-lactamase inhibitors. So currently in development, I'm aware of 4. I say aware of 4 because you just never quite know what else is going on, but I'm aware of 4 different combinations of beta-lactam and beta-lactamase inhibitor, and as I mentioned before, meropen and vaborbactam is in its latter stages of review with the FDA. It's a slightly unusual one in that it's what's called a cyclic boronate, which gives it a structure that is likely to be more difficult for the bacteria to circumvent. Time will tell. Fingers crossed. Imipenem silostatin has been around for 30-something years. Merck have now added relebactam to that to protect it against a wide range of beta-lactamases, as trianam, avibactam, and sulbactam, and diazobicyclinate are other combinations that are in development. But, and this is the piece that's particularly relevant, is that gram-negative species such as Pseudomonas, Acinetobacter, both of which were in the top 3 nasty bugs from the WHO, they have other ways, additional ways of overcoming beta-lactam antibiotics. So they can institute porin change, effluxes. I'll talk about those in a moment. So one of the other ways is to actually try to get your antibiotic into the cell on the back of an essential mechanism for the cell. So what's affectionately called the Trojan Horse approach. And for those of that don't remember your Greek mythology, I suggest you go and have a look at the Trojan Horse story at some stage. And then towards the bottom of the slide here, one organization is actually looking at 2 different molecular approaches, but looking at how to block the activity of efflux pumps so the bacteria can't pump out the antibiotic as it enters, and then to try to manage porin changes such that antibiotics can penetrate the cell. So there's some creative ways of looking at beta-lactam resistance. This is the Trojan Horse approach, and hopefully you can follow this through. I was going to try and use the arrow, but I'll not for the moment. So there's an ion transport system. As many of you probably realize, bacteria require ferrous and ferric ions for normal maintenance, normal function. It's a critical ion. And a molecule called siderophore, which is a catechol in this case, stuck onto the side of a cephalosporin, actually binds to the iron ions. And because the bacteria needs that, it sends out scavenger molecules to find the iron, and by design pulls in the antibiotic as well. So you've got this iron complex with an antibiotic complex, and it's actively, physically drawn into the bacterial cell. Once in the cell, the 2 split, the antibiotic is released, and it goes about its merry way and attaches to the penicillin binding protein, which are those funny green things you can see at the bottom of there. The other approach is that porins, that I mentioned already, these are the portals of molecules in and out. Again, you can, the Trojan Horse will allow access of these particular molecules without any issues, and the same applies to the efflux mechanism. There is the way that the Trojan Horse works is that it actually avoids, because of the critical nature of the iron, means that it doesn't get effluxed. The other component is what the antibiotic that's actually released is resistant to many, not all, but many of the beta-lactamases. So the Trojan Horse is a fascinating 3-armed way of overcoming some of the problematic multi-drug resistant pathogens. Okay, so where are we now? Well, there's a myriad of different ways of trying to look at to develop new antibiotics. I'm going to give you some examples of most of these, and I think it's a mark of our ingenuity and our approach to this growing challenge that we're really trying to look forward, but also look back. And I'll go through some of these and give you a feel for how I think we're actually making some progress. One of the key things that I think is particularly important is that companies, academia and governments are actually sat on vast amounts of information. Just think of the amount of information that gets submitted to the regulatory authorities, and the drug may or may not be approved, but there is this mass of data that's sat there, and it may well be of value for future developers. So the Pew Trust, which is a charitable organisation based here in the United States, has actually put together a global data platform whereby they're pulling together all of these different information sources and making them searchable, shareable, so that new people coming to the field that aren't aware of research that was completed 10 years ago, certainly 20 years ago. I think many of us are not as old as me that would not be aware of some of the research that was completed in the 1980s and 90s on quinolones or on tetracyclines. So this global data platform is a novel way of trying to bring all of the information together in a true sort of big data approach. And I think information sharing is one way in which we can avoid reinventing a wheel, especially if it wasn't a good wheel in the first place. So that's the Pew Trust approach to this challenge, and this is what their website looks like. It's called Spark, and it's pretty user-friendly. Even I can use it. And what it does, you can see there, it looks at the chemical structure, provides an array of different chemistry information, such as any HTSM results that were put through. And HTS was very much in vogue in the 1980s and 90s. It's not quite so in vogue nowadays. We have different approaches, but there is a huge amount of information there. Biological information. It saves having to go through lots more MIC testing, animal models and so forth. An awful lot of molecules have to be registered for patent reasons, so those are now on public databases and they're accessed here. And obviously, as molecules were developed, there's expert commentaries, there are published articles on their activity, the pharmacokinetics and so forth. And it actually, this Spark pulls this all together so that people will know who is actually looking at what and how things are going to move forward. So it's a genuine effort to be comprehensive and open and collaborative. Future discovery options. There's a variety of ways in which we could tackle this. I could speak for way too long about this topic, but I think there are three components here that I think are particularly important. There's this essential gene paradox, whereby we know that just a small proportion of the genome of E. coli, which is around 300 genes, are critical for growth. But there are 250 or so that are required in certain conditions. So are we able to target some of these previously unrecognized genes and actually create them as targets for antimicrobial therapy? And some of these candidates include the bacterial response to stress. We've known about that sort of stress response really since the late 80s, 90s with some of the quinolone work. Type 3 secretion, which is the second one here. This is being investigated in a gram-negative setting, and it's really quite a clever way in which bacteria can attack host cells. Attachment and nutrients are other ways in which we may be able to identify certain essential genes and conditional genes in order to come up with different target sites. And then there's the gene-protein interactions, which an awful lot of these interactions are actually highly complex and fairly redundant in many cases. Can we utilize those interactions in which to institute some form of lethal action? Chemical genomics is a vast topic in of itself, and one of the first companies I'm going to speak about actually is doing some quite elegant work in that area. And then combinatorial discovery. That is an area whereby we can, or are, I should say, looking at old drugs with new additions to sort of recreate a new combination, but using a compound that we've known and used for many years, but really just sort of supercharging it, for want of a better term. So those are some of the future discovery options, but what I'm going to do now is run through some of the different approaches to the antibiotic resistance challenge. And This is by no means a comprehensive list of all of the different companies in this space, but these are the ones that I thought helped go to some of the points I've been trying to make about the novel ways in which we have to tackle the problem. And the very bottom line is one that I think will be intriguing to many of you as well, so I'll leave that for the moment. A UK company called DISCUVA have two different what they call platforms. One is called SATIN, which, as you can see, is selective antibiotic target identification. And what they're doing here is they're looking at and adopting something like the Pew approach. They're looking at an array of different antibiotic classes. Some of them failed, some of them are current compounds that could do with a little bit of zip to make them more potent. And they're looking at a range of different pathogens simultaneously. So if you think about the different pathogens, the different compounds, that's a huge matrix of information that needs to be assimilated. And they can also look at the different pharmacokinetic and thus pharmacodynamic components with regard to certain antibiotics. Can we reinvent an old antibiotic by giving it once a day but a higher concentration, or things like that? I'm sure you've got the drift of where I'm going with this. So one of their platforms is called SATIN, where they're trying to pick out certain antibiotic targets. And as you can work your way around the wheel, you can see that, we know the mechanism of action. We know how the compound goes into and gets pushed out of the bacteria. We can look at, or they can look at, how to fine-tune this efflux mechanism that I described. And we can look at how resistance develops. Is it a slow process, or is it a rapid single-step process like you would see with rifampicin, for example? And obviously, with the chemistry capabilities of today, you can do structure-activity relationships and assess what the effect of certain molecular changes would be on efflux or entry into the cell. And then there is the overall toxicity that's associated with different molecular classes. We know some are associated with certain structures, others we only find out when we've exposed them to humans. So that's SATIN. And then we have, and this is how the process works in terms of a library creation, creating mutants, and then the mutants are assessed against an array of different chemicals and potential antibiotics. And that's a list of some of the antibiotic organism libraries around the wheel at the bottom: E. coli, Staph aureus, Neisseria gonorrhoeae, and so forth. So you can see they're homing in on the WHO's list of priority pathogens. This is the program called SILK that I was just about to start to describe, and this employs big data, classic big data, huge amounts of information. It's assimilated. They look at screening processes, they look at mutation rates. Do mutation rates change in different conditions? We know that the pH in infections is lower than it is in normal healthy tissue. So does that have an effect upon mutation rate? That's the sort of thing that SILK can address. It looks at serial and compound mutations. So you can normally, in the past, subjected organisms to multiple repeated passages to an antibiotic and waited for resistance to emerge. This sort of mechanism can actually predict these types of things. So as you can see, all of this information is combined to look at multiple candidates simultaneously against the types of organisms that were shown on what I'm going to call the SATIN wheel before. And I think this sort of big data and genetic manipulation is the right sort of way to pull out more focused candidates for future research. I'm going to go into what I'm going to call the natural world now. You may recall, going back to the wheel of different ways to bring in new antibiotics. Well, Agile Pharmaceuticals have actually gone underwater, literally underwater, and they have, using molecules produced by marine organisms, and those molecules are there to protect themselves. One of the problems of being underwater is that you can be covered in biofilms, and the biofilms are produced by unpleasant bacteria and so forth, and destruction of sponges and sea squirts and so forth. And these molecules may well have important effect against things called biofilms, which are intrinsic in urinary tract infections, bone infections, heart valve infections, biofilms exist almost anywhere that there is an infective process. So the Agile folks actually worked from the compound agileferin, and have been manipulating the molecule, adding different components like bromine and other atoms to different parts of the molecule. And they've done it to such an extent that they have patented and published a wide range of different molecules that are significantly more antibacterial than the original molecules that you saw a few moments ago that were recognized in the late 1980s, early 1990s. And it is this chemical sophistication that is going to enable a natural product from under the water to actually be of potential value in things, as I say, like urine infections and so forth. So there we're going to a natural resource that we haven't been to previously. I think we're all familiar with the sort of the cephalosporin story of the Italian gentleman sifting through sewage to find the right fungus that produced the cephalosporin. Well, we're going to a different source now. We're going to the algae and the biofilm organisms. And it's proposed that these algiferins, they've got a novel mode of action. It's not been seen by the bacteria that we struggle with, and they have what's called a two-component regulator mechanism, and the algiferins disrupt those. They clearly disperse biofilms, which is a great step forward, and they appear to actually restore activity of some antibiotics against the pathogens that we're worried about. Maybe they can mitigate the acquisition of resistance. Time will tell. The bacteria are very, very smart. Cincer, another very, very small company, are actually working on non-natural, those synthetic antimicrobial peptides. And these peptides work, they're only 8 to 10 amino acids in length, and they can be built, literally built, such that they can be tailored for certain species, specifically certain species, which is very much the narrow spectrum approach of antibiotic stewardship. So this is obviously a good way to go. And what happens is the small string of peptides gets in, and you can see here that the big circle shows you the lipopolysaccharide, the O antigen as it's called here, linking to the lipid in the outer membrane here of gram-negative bacteria, and the peptide, the nanomolecular peptide, actually inserts itself into both the O antigen chain and at the face of the O antigen binding to the lipid A, and it basically just cleaves it and bursts a large hole in the cell membrane. And as you well know, you've got a hole in bacterial cells and they just burst. So if you look at the two black and white pictures, you can see on the top there's a typical image of a bacterial cell, nice and fat and rounded, and then one below where they've been exposed to the spearhead antibiotics, the cells are just lysed. So I think that's an interesting way of tackling the gram-negative problem. I believe that there's also work going on to look at gram-positive bacteria in a similar sort of way. So we see where the spearhead technology takes us over the next few years, but this is an interesting approach to which bacterial resistance may be slower than some of the other molecules we've seen previously. Cursor Pharmaceuticals have looked at the ribosome, protein synthesis. Now, you may recall that the macrolides work at the 50S subunit of the ribosome. Tetracyclines work at the 30S ribosome. These clever people are going to a completely different part of the ribosome called the P site or the 70S site. And this is a novel ribosomal target site. And because of that novelty, it's clear there's a new mode of action involved here. And it means that organisms that are resistant to the macrolides and other similar classes of antibiotic resistance are overcome by virtue of going to a completely new site. And if you look at the center of this particular image, you'll see that macrolides, lincosamides, chloramphenicol, they work in the 50S component. Plurimutilins, which is Lefamulin, you may have heard me mention that previously, that also works in the 50S, but at the lower end. Whereas here, the cursor molecule works in a completely different part of the protein synthetic mechanism of the ribosome. And I think that is it's going to be an interesting development program, and we shall wait and see. So far, the in vitro, and I stress the in vitro activity, suggests that it may work with pathogens as broadly as Mycobacterium tuberculosis through to multi-drug resistant gram-negatives like Pseudomonas. Novobiotics, they actually got two platforms, so I guess they're hedging their bets. And one of them, Cystamine, actually has been used for quite some time as a well-known agent for a nephrotic condition. Move along. And the cystamine is actually an endogenous immune modulator. And we know that actually if you add cystamine to an antibiotic, it improves the activity of the relevant antibiotic, whether it's quinolone, an aminoglycoside, or whatever. So the combination of cystamine, a compound, as I say, that has been in clinical practice for probably about 30 years in a rare disease, in a rare condition, but nevertheless used significantly and safely, major issue there, over many years. And if you could add that to an antibiotic that is being established, as I say, in aminoglycoside and so forth, you could actually reinvigorate. And that was one of the other squares in the how to develop new antibiotics, repurposing, reinventing an old drug. And in fact, there is a study ongoing looking at cystamine with an antibiotic. It's actually an aminoglycoside, in the study of cystic fibrosis. Now, cystic fibrosis is not a common infection, or it's a common condition, but infections can be life-threatening. And certainly over the period of a lifetime of a CF patient, the repeated infections really are a significant problem. It's also been shown that the cystamine with an antibiotic can disrupt biofilms, which again goes to the cystic fibrosis component and will also contribute to useful clinical activity in complicated urine infections and possibly things like ventilator-associated pneumonia, where the pneumonia develops really from a biofilm developing on the inside of the ventilator tubing. The other platform that Novobiotics are looking at involves engineered peptides, and again, it's a little bit like the SINSA program. They can actually engineer the peptide to specifically target the cell membrane of the bacteria and also fungi in this particular instance, and they describe a starburst activity on the membrane and rapid lysis, and they seem to be able to build the AMP, as it's called, the peptide, for each pathogen, as I mentioned before. It's as though the pathogens have got a unique lock and key. So that's Novobiotics. Quorum Therapeutics are looking at quorum sensing and how to interfere with quorum sensing, because that's the way in which the bacteria recognize there's something going on or an opportunity to infect, and it can control virulence. And this complex picture shows you what's called an accessory gene regulator that switches on the production of certain virulence mechanisms and toxins. And the folks at Quorum Therapeutics have developed a small molecule that actually blocks a critical part of this quorum sensing pathway, and it blocks what's called the AGRA component. And by blocking this component, this pathway, it should be that we reduce or completely eradicate the toxic effects of certain bacteria. And here they've been looking at Staph aureus. So this is a new way to affect the virulence, and that should ameliorate the effects of the disease and maybe even prevent the disease. Microbiologist is a company that's focused on two things. One is type 3 secretion, and this is where bacterial proteins are actually translocated or injected. And here you're looking at a syringe sort of upside down, and where all the different circles are at the bottom. That's where the protein is manufactured, and it's injected up into the target cell, the host cell. And we know that the Pseudomonas type 3 secretor will kill a whole range of different immune protection cells, such as macrophages and neutrophils. And type 3 secretors are known to be associated with poor disease outcomes. You heard me talk earlier about efflux pumps. This is a very complex efflux pump called the RND, and RND can confer a wide range of different resistances to different agents and indeed here biocides. And one of the things that this company is looking at is how to inhibit the way in which these efflux pumps work. And the potential benefits are significant. Coming towards the end here, antibiotic resistance breakers, and I acknowledge David Brown, and his paper is well worth a visit if you have the chance. And what he's done is, and other workers have done, they've looked at compounds that we've used for years: berberine, cycloperox, loperamide, one of the classic antidiarrheal drugs. All of these have got some activity either on the immune system or directly on the bacteria themselves. Cycloperox works on inhibiting lipopolysaccharides in gram-negatives, and by the way, it can block some of the immune reactions. So each of these listed here have got some potential in terms of interfering with the resistance process, either direct antibacterial activity, they can improve other antibiotics in terms of resistance, or they can actually clear the infection, eradicate the infection by blocking some of the host responses that can lead to the inflammatory process. So, just to show you the ingenuity of microbiologists and where they look for the next penicillin, a couple here that caught my eye. One is the Australian researchers that actually found in the Tasmanian devil milk, so clearly, not an easy source to get at, but milk from Tasmanian devils would actually kill MRSA. But as it says here, milking Tasmanian devils is not an easy process. And then there is another group that have actually found that there is a novel antibiotic that's been found in the nests of leafcutter ants. So our ingenuity is appearing to have no limits, and I hope that whether it's Tasmanian devil milk, leafcutter ants, or some of the ingenious mechanisms that I've just been describing actually provide us with new weapons against antibiotic resistance. Thank you for your attention.
Thank you so much, Dr. Tillotson, for that informative presentation, and thank you so much for your research. We will now start the live Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of your presentation window, type in that question in the box that appears on the screen, and click the send button. We'll answer as many questions as we have time for. So let's get started. Our first audience member has a question. Dr. Tillotson, how long does it take the identity to identify a drug and get it approved?
Okay. It takes anything from 15 to 20 years for an antibiotic candidate drug to be identified, studied in the clinical trial programs, and then be approved by the regulatory processes. But it's important to note that not every drug that goes into that process makes it all the way through. So anything from 15 to 20 years. We are getting quicker, but it still takes a long time.
Thank you so much. And here's our next question. We have a lot of wonderful questions coming in. How many candidates have to be tested to arrive at a few possible agents?
Typically, we would say that 5,000 candidate molecules would lead to a single viable compound. And a lot of those 4,999 are excluded for reasons of poor activity, toxicity, the pharmacokinetics are not ideal for human administration, and so forth. So the attrition rate is quite high. And I think it's higher in antibiotic development than in other branches of pharmaceutical research, because other branches work with Homo sapiens. Here, we're working with Homo sapiens and a third party, the bacteria or fungi. So it puts another layer of complexity onto the process of finding a candidate and then putting it through its paces. So 1 in 5,000 is a typical sort of hit rate.
Thank you, Dr. Tillotson. This is such important information. Here's another important question from one of our audience members. What does it cost to develop a new antibiotic?
I would say with today's technologies and the more focused approach that we had, say, 20 years ago, I would say that it can cost anything from 500 to 750 million to bring an antibiotic to the clinic. Now, part of that obviously is factoring in the costs of those 4,900 or so drugs that didn't make it, but nevertheless, to actually take an antibiotic through all its paces to meet the regulatory requirements and so forth is a minimum of $500 million.
Thank you, sir. We have time for one last question from our audience members, and it looks like we have several. Just a reminder, go ahead and send them through and we will get to you as soon as possible. Why does it seem to be only small companies in this field, Dr. Tillotson?
Yes, that's a very good question. Without wishing to be cynical, I think a lot of the large pharma companies have, how can I put it, stepped back from some of the research initiatives that they had 20, 25 years ago, mainly because of the challenges of bringing new antibiotics to the clinic. I think at the moment the small companies have the field because they are compact, they are responsive, they are not the huge megalithic organizations that some of the other companies are. And I think many of the companies that I've referred to in my presentation are literally less than 20 people. In fact, I know 2 companies that are literally 3 and 4 people. So they're a lot more flexible. They can adapt to ideas and to challenges much more rapidly. And I think once a drug is looks like it's being proven, looks like it's living up to the hype, then I think the big companies step in and take the drug through its last stages with FDA and EMA, and that's when the big bucks are needed. That's when you need the $85 million to do 1 clinical trial. That's when the big guys come in. But a lot of the early innovation is driven by the small companies, and in fact, a number of those initiatives actually are coming from universities and other academic settings where someone has a bright idea, it gets turned into a small company, and it grows from there. So I think innovation and flexibility are the 2 hallmarks of a small company, but the sheer mechanics and investment comes from big pharma.
Thank you, Dr. Tillotson, for your presentation. Before we leave, audience members, thank you so much for being here today. Dr. Tillotson, do you have any last comments you'd like to share with your audience today?
Thank you, Susie. I think I started off on a note of true pessimism that we managed to destroy Fleming's work within 70 years. I think with the awareness of how to use antibiotics more appropriately, both clinically and economically, and a lot of the innovation that's driving research ahead at the moment. I would hope that in the next 70 years we actually see this trend reversed. I'm not naive enough to think we're suddenly going to see a wave of new antibiotics, but I think people are now aware of what's happening and this impending tsunami of resistance. So I don't want anybody to think that we've got the Armageddon bacteria around the corner. I think we have the science, the skills, and the tenacity to actually turn this thing around. And I look forward to the next 10, 20 years. So thank you.
I would like to once again thank you again, Dr. Tillotson, for your presentation. I'd also like to thank LabRoots today for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again. Bye-bye.